Hello, recruit, and welcome to Astroneer Academy 205, Xenobiology. Everyone knows that your adventure as an astroneer is a lot more fun with friends. Wait, no, not these friends. I am talking about these friends. While that first group of friends can be a lot of fun, they can also be a liability. They'll take all of your QTRTGs to power hoverboards, use all of your compound to make needless tethers, and maybe even accidentally blow up your entire base. This other group of friends won't do any of those things. In fact, each of these adorable little friends have unique perks that can offer to assist you in your adventures as an astroneer. Today, we will begin the rather long mission chain that introduces us to each of these friends and discuss some best practices and pro tips along the way. Xenobiology is a rather involved topic, however, so we have broken it down into two parts. Today's course will introduce the first of these friends, with next week's course introducing the other six. So, let's get started. I suppose we should begin with a question. What is xenobiology? Let's turn to Wikipedia for the answer. Xenobiology is a subfield of synthetic biology, the study of synthesizing and manipulating biological devices and systems. The name xenobiology derives from the Greek word xenos, which means stranger or alien. Xenobiology is a form of biology that is not yet familiar to science and is not found in nature. So essentially, xenobiology is the study of synthetic or man-made life forms, with its root word xenos pointing to possible alien sources. So all of these little cuties are alien life forms? Well, maybe. I suppose we will find out over the course of the next 2 weeks. We now know what xenobiology means, but what type of alien or synthetic life form are these adorable little creatures? These, recruit, are Galastropods, also known as Space Snails. Their very existence was the stuff of rumor and legend for ages until, one fateful day, Exodynamics sent a new mission chain to our mission log that would help us begin to confirm the identity of all 7 space snails, unraveling the mystery of their existence, and take us on a journey across all 5 planets and the 2 moons of the Sylva planetary system. We actually completed the first of many xenobiology missions back in Astroneer Academy 202 when we collected 3 sphalerite. If you have not yet done so, you can head down into the caves of Sylva or the surface of Desolo to gather sphalerite to complete the bait and switch mission. If you do not see this mission available in your mission log, be certain you have completed the Smelteringly Hot mission. Once you have completed bait and switch, you can claim your reward from the mission log on the nearest landing pad. That reward is this. This tiny purple object seems to be made of the same stuff we will see again in gateway chambers, and when you hover your simulation cursor over it, the hologram is absolutely enormous. Check out that description as well. This strange object speaks of Chronos case files and the IS Triton. What is this all about? Well, we'll get to it, recruit. Be patient. Completing bait and switch opens up a new mission, Strange Object. This is a very easy mission to complete, but there are a few things to be aware of. To complete the mission, you simply need to place and activate the strange object on the surface of Sylva. When activated, the strange object will open what appears to be a fairly large Einstein-Rosen bridge, through which will appear a wrecked Xenobiology Lab that was jettisoned from the ISS Triton. I'll bet that last sentence didn't really make much sense, so let me break it down. An Einstein-Rosen bridge is the proper term for a wormhole, which is a feature in space-time that would essentially provide a shortcut between 2 points in space that would otherwise be separated by millions of light years. You will see them again and again in your adventure as an astroneer. As for the ISS Triton and the reason the Xenobiology Lab was jettisoned, well, we'll get to that next week. Since the Xenobiology Lab is fairly large, you need a rather large, clear area to deploy it. I should also point out that you can only activate the strange object on Sylva. If you take it to another planet and try to activate it, you will encounter an error. If the area around the hologram is not clear, you cannot deploy the Xenobiology Lab. There's a nice visual indicator to help guide you. When the area is not clear, the hologram remains mostly an outline of the Xenobiology Lab, but once you move the strange object to an area that is clear, the hologram takes on a noticeable shimmer. Before you activate the strange object though, be aware that the Xenobiology Lab will flatten a small amount of the surrounding terrain on the same plane the strange object is resting. That means if the strange object is resting at a slight angle, the surrounding terrain will be flattened to that same angle. To help prevent any bizarre placement, consider extending a bit of your flat surface from your base over to the area you wish to deploy the Xenobiology Lab. This will ensure it is nice and flat when you deploy it, and it will even have it on the same level as your main base. If you look closely at the hologram of the Xenobiology Lab, you will notice that one end of it is smaller than the other. If you rotate the strange object while holding it and align that smaller end so that it's on your left with the larger end to the right, then the control panel and other key portions of the Xenobiology Lab will be facing you when you activate the strange object. You can deploy the Xenobiology Lab facing any direction you wish, but knowing which side the controls are located on before you deploy it can help you make an informed decision about its placement. Once you have located the location you wish to deploy the Xenobiology Lab, place the strange object on the ground and interact with it. The Einstein-Rosen bridge will open just enough to make room for the Xenobiology Lab, causing it to appear where the strange object was. The surrounding area is flattened, and several pieces of debris will appear nearby. This will complete the strange object mission, and you can collect your reward of a power cell from the mission log control panel of your landing pad. Let's take a quick tour around the Xenobiology Lab. On the far left end, you will find a control panel, a single small Tier 1 attachment slot, and a power cable plug. Just above that is a radio dish that was apparently used for communication or data transfer for the occupants of the ISS Triton. Just to the right of the power cable plug are a handful of displays that are currently unpowered. Amongst these displays is a power indicator. It is the horizontal, oval-shaped section with 4 segments. To the right of that, in the larger portion of the structure, you see an open storage bay that features a single medium Tier 2 slot and a hologram of an oxygenator. Directly above that, spanning the top half of this large rounded section, you see additional storage bay doors that are currently shut. As you move around the Xenobiology Lab, you will see quite a bit of debris that is most likely partially embedded in the surrounding terrain. You can use your terrain tool to access this debris if you wish. Like all debris, it can be placed in a shredder and turned into scrap. Around the back of the far right side of the Xenobiology Lab is a much larger piece of debris that almost appears as though it may have once been part of a section of a spaceship. Inside, you will find a fully functional oxygenator. I bet you can guess where that goes, along with various resources. Completing the Strange Object mission will also introduce 2 new missions, a breath of fresh air and jumper cables. You can complete these 2 missions in any order you wish. If you read the description of A Breath of Fresh Air, you discover that the Xenobiology Lab needs to be reoxygenated for safety and that you should investigate nearby debris to locate a functioning oxygenator. You can retrieve the oxygenator from the large debris behind the Xenobiology Lab and place it into the open bay where the oxygenator hologram appears. Note that once you place the oxygenator into the Xenobiology Lab, it cannot be removed. This will complete the current mission, and you can collect your reward of 250 bytes from the mission log on the landing pad or from your action wheel. The next mission, Jumper Cables, is equally simple. You need to recharge the internal power of the Xenobiology Lab. You can go about completing this mission in a number of ways. The most obvious is probably the power cells you received as a mission reward moments ago. You definitely can place that power cell into the small Tier 1 slot on the Xenobiology Lab, and it will fully recharge the internal power. You will, however, find yourself recharging that internal power numerous times during the Xenobiology mission chain, so you may wish to consider something a bit more permanent. If you deployed the Xenobiology Lab in close proximity to your main base, you can simply use power cables from a nearby platform or utilize extenders to connect the Xenobiology Lab into the power network of your base. If you deployed the Xenobiology Lab further away from your base, you can also simply connect a small power producer directly to the power cable plug on the Xenobiology Lab, or you can connect a platform and store a larger power producer on that platform. Regardless of how you choose to recharge the internal power of the Xenobiology Lab, doing so will complete the Jumper Cables mission and reward you with 250 bytes. It will also open up the next mission, Know Thy Galastropod, which is the first in this mission chain to feature the unique Galastropod icon in the mission log. To complete this new mission, you simply need to interact with the Xenobiology Lab's control panel and press the big green button to activate its tracker. Doing so will allow you to collect a mission reward of an oxygen filter, but it also opens up a small number of entries on the data log contained in the control panel of the Xenobiology Lab. The information contained in the data log will begin to tell the story of Dr. Ines Yo and her assistant, Now. We will discuss a summary of what we learned through the personal files of Dr. Yo next week, so for now, we will only discuss what we can learn from the Snail Data and Specimens information in the data log. We will cover each entry as it becomes relevant in our discussion this week and next week. First, we need to investigate the next mission, G. Sylva Shells. This mission tasks us with locating and scanning 5 discarded Galastropod shells in the forest of Sylva to provide data to the Xenobiology Lab to aid in uncovering the Galastropod specimen located on Sylva. You have most likely already encountered these shells, as they are fairly abundant in the forest region. When you do encounter one, simply interact with it to scan it, which will also provide a small number of research bytes. Once you have scanned 5, the mission will be completed and you can return to your landing pad to claim your mission reward of an empty terrarium. Yep, it's empty, but our next mission, G. Sylva Terrarium, provides instructions on what we need to add to it in order to create a comfy habitat for a new friend. The terrarium itself is a small Tier 1 item that features a small Tier 1 slot on the top, and the terrarium can stand on its own included support legs when placed on the ground. It can also be attached to any Tier 1 storage slot as well. I want to backtrack to the Galastropod shells for just a moment. While the mission objective only requires you to scan 5 of them, you can continue scanning them again at any time you wish to obtain bytes. You can also use your terrain tool to dig them up so you can place them in a research chamber where they will yield a higher number of bytes than just scanning them, or you can place them in the shredder for scrap. Additionally, if you are encountering difficulty locating discarded shells to scan, simply open your compass while the shells missions are active. The 3 nearest shells will appear as nav points on your compass, making it easier to locate them. If there are additional steps required before you can scan the shell, the nav points will appear grayed out. You can refer back to Astroneer Academy 106 for further information on utilizing your compass. Now, back to our current mission. Investigating the mission objectives reveals that we need to fill the terrarium with soil, zinc, and a bounce vine seed. You can collect these items and place them in the terrarium in any order you wish, but we will begin with the bounce vine seed. You are introduced to the bounce vine in Astroneer Academy 102, so you know that they can be found in the mountain region of Sylva. Simply dig up a bounce vine, pick up one of its seeds, and place it on top of the terrarium. The seed will be consumed, and a habitat will begin taking shape inside of the terrarium. There is a segmented ring around the Tier 1 slot on the top of the terrarium. At this stage, this ring indicates your progress in creating a suitable habitat for a Galastropod, with each required item filling up 1/3 of the ring. The zinc should be easy enough to obtain since the bait and switch mission that kicked off this whole Xenobiology mission chain required you to collect 3 sphalerite. Refine one of those sphalerite into zinc with your smelting furnace and place the zinc on top of the terrarium. Now we just need a bit of soil for the terrarium, which is completed by simply placing a soil canister on the Tier 1 slot of the terrarium. Okay, so not just any soil canister, it needs to be a soil canister that actually contains some soil. There is some debate amongst seasoned astroneers as to whether that soil canister needs to be full or not. Your experience may vary, but some astroneers have had success completing the mission by supplying the soil canister with a rather negligible amount of soil in it. Others insist the soil canister must be completely full in order to satisfy the mission. Personally, I use a full soil canister since it's incredibly easy to refill a soil canister anytime I wish. Whichever approach you prefer, be prepared to retrieve your soil canister once you have attached it to the Tier 1 slot on top of the terrarium. At times, the soil canister will simply empty its contents into the terrarium and then remain attached to the Tier 1 slot. You can simply pick up the soil canister and place it wherever you wish. There are other times when the canister may be ejected from the terrarium, often with a fairly impressive force, sending it flying into the air. This seems to happen more frequently when the soil canister was the final mission requirement item that you supply to the terrarium, though I have seen it happen at other times as well. If you have good timing, you can simply catch the soil canister while it's still in the air. If not, pay attention to where it goes so you can pick it up and continue using it. With all 3 mission requirements met, a comfy little habitat is now ready for a Galastropod, and you can also claim your mission reward of a small trumpet horn. This mission will also unlock the schematic for the small trumpet horn in the research catalog, so you can create more if you so desire. We will talk more about all of the various musical horns and how they can be utilized with storage canisters in an upcoming course sometime next quarter. We are now ready to complete our newest mission, G Sylva Recovery, and meet the first of the 7 Galastropods. Before we take this big step though, let's consult the data log of the Xenobiology Lab to learn a little bit more about Galastropods. The first entry under the Snail Data section is an eye-opener. While we do not know where Galastropods first appeared, we learn that they have arrived in the Sylva planetary system by way of their eggs hitching a ride on comets. So that answers a question from earlier in our discussion. Galastropods are, by definition, aliens. Worse, they are an invasive species, which means they are actually alien invaders. Who knew these cute little things could actually pose a massive threat to the ecological balance of every planet they arrive upon? Once hatched on solid ground, they are quick to adapt to their environment, resulting in a variety of features that help them survive. In the Evolutionary effects entry, we learn that Galastropods have evolved the ability to, and I quote, hijack a neurological response in the orbitofrontal cortex of other beings. While the orbitofrontal cortex of our brain is responsible for many things, it plays a role in modulating visceral reactions, which are powerful physical responses that can occur when something is perceived as threatening, joyous, dangerous, or other strong emotions. So essentially, the data log is telling us that the Galastropods are able to make us say, "Aww," and want to care for them. You know, this. On the upside, each Galastropod provides you with different positive effects, so we do get something out of the deal. The very next entry helps us understand how we can befriend Galastropods who are known to be playful and mischievous. Pay attention to the clues about music, dancing, and interaction, as all 3 will become relevant in just a few moments. Finally, we learn that Exodynamics recommends keeping the Galastropods in their terrariums to not only ensure their safety, but to prevent them from wreaking havoc on the local ecology. Remember, we are dealing with an invasive species and, as such, they could easily upset the delicate natural balance of any planet if they were left to their own devices. There is one other trait of Galastropods not mentioned in the data log that relates to a special vehicle we discussed near the end of Astroneer Academy 106. Hold on to that thought because we will come back to it in just a few minutes. The final item I want to bring to your attention before we complete the current mission is the Specimens section of the data log. Most entries are currently encrypted, so we cannot read them, but we can learn a bit about the space snail we are about to meet. Galastropoda Sylva, also known as Sylvie, is a brave and curious galastropod who emits light from their body when well fed. We're not talking about the light from 10 million fireflies who fill the world as you fall asleep. But I do believe the light will still cause you to stop and stare. So let's do this. Let's see if we can't bring Sylvie out of hiding, befriend them, and complete another mission. To get started, you need to attach a small horn to the top of the terrarium. You can attach the horn while the terrarium is stored in your backpack or other tier 1 slot, or while it is freestanding. The result will be the same. You can use the small trumpet horn you received earlier, or you can use a small squeaker horn that can be found on discarded backpacks. Either one will cause the terrarium to begin playing a tune. As it does, your attention will be automatically drawn to Sylvie as they come out of hiding to investigate the music. You will receive a notification that your compass now has a nav point indicating the location of the Galastropod. While this is not all that useful when a space snail first appears, since you should now be staring right at them, it can come in handy after your first interaction. You may encounter an error when placing a horn on the terrarium that states no Galastropod can be found in the area. If that happens, simply remove the horn, wait a brief moment, then reattach the horn. This will typically result in successfully luring Sylvie out of hiding. If you continue encountering the error, simply move to a slightly different location, then remove and reattach the horn again. You don't need to go far, just a few steps in any direction is typically enough to resolve this error. If you placed the terrarium on the ground earlier, I would recommend placing it back into your backpack at this point. The tune will continue playing, and this will make befriending Sylvie a bit easier in just a moment. As you approach Sylvie, they will wave to greet you, and you'll receive a prompt to use the emote wheel to dance with the Galastropod. We will be covering the emote wheel further in an Astroneer Academy 301 extra, but for now, pay attention to the part of the prompt that calls out performing a dance emote specifically. Other emotes will not work for our purposes today. You can dab in front of Sylvie all day long and they will not be impressed. Of course, you can dab in front of me all day long and I wouldn't be impressed either. Who still dabs? Anyway, you need to perform any of the dance modes, and when you do, Sylvie giggles and runs away. Before they run away though, you may notice the marker that appears in the simulation just over their head has a circle that fills halfway. That circle is an indicator of your progress in befriending a Galastropod and will appear for each and every interaction in the recovery mission for each snail. That brings us to a quick pro tip for befriending the snails. After you perform your dance, the snails will run off to a nearby location. You can use your compass to locate them, or you can just look around and see if you can spot the marker that gives away their location. But you can also pay attention as they burrow into the ground and watch for telltale dust clouds to appear just above the terrain. These small dust clouds indicate the direction the snail is burrowing underground and will help you look in the right direction as they emerge back on the surface. Approach Sylvie for a second time and they will once again giggle because snails are just playful like that. Perform a second dance for Sylvie and they will become visibly excited and then begin singing and dancing along with the tune playing from the terrarium. At this point, you can remove the horn from the top of the terrarium and place it near Sylvie. When you do, you will notice the prompt to befriend Sylvie. Hold down the input indicated on the prompt, and Sylvie will automatically relocate into the terrarium, bringing an end to the tune it was playing. This will also complete the G Sylva recovery mission and reward you with, well, a new friend, Sylvie. Unlike other missions we have completed so far, the recovery missions for snails do not archive in the completed section of the mission log. Instead, they appear in a new category, Reclaimable. As the name implies, you can activate this entry in the mission log to reclaim Sylvie at any time you wish. When you do, Sylvie will be retrieved from their current location and placed on the landing pad you used to reclaim them. You can even reclaim Sylvie from your own backpack. Go ahead, give it a try. It's a pretty cool gimmick. You will see other items appear in this reclaimable section as we progress through various mission chains, and they will always function essentially the same. Completing the recovery mission introduced the G Sylva verification mission. To verify Sylvie's species, they must be scanned by the Xenobiology Lab. This process is incredibly simple. All you need to do is place Sylvie's terrarium into the small Tier 1 slot next to the Xenobiology Lab's data log. When you do, your attention will quickly be drawn across the front of the lab. You will see the small round screens illuminate, and your focus will come to rest on a small chamber behind the newly opened door. You will be able to watch as Sylvie is scanned, after which you will once again be in control of your point of view. This will complete the verification mission, so you can remove Sylvie's terrarium from the slot on the lab and claim your mission reward of 250 bytes. You can now store Sylvie wherever you wish, or simply carry them around in your backpack. 2 new missions have opened up: Galastropod Care and Tracking Power. We will tackle Galastropod Care first. All snails can provide unique positive effects that can aid you on your adventure. How long that effect lasts will depend upon what you feed the snail. But what do space snails eat? A quick glance at the mission objective will reveal that space snails like to eat the seeds of the various hazardous flora. Any seed will do, even those from other plants, but each snail has a favorite food. If you hover the simulation cursor over a space snail's terrarium, the tooltip that appears will list their favorite food. Sylvie prefers mutant hispine seeds. I can hear you thinking, if any seed will do, why do we need to know their favorite food? That's a great question, recruit, so let's talk about it. Feeding a space snail any seed will activate their indicated ability for 10 minutes. If you feed them their favorite seed, however, that ability will become activated for 30 minutes. For the most part, when their ability is active, the ring around the top of the terrarium will have a blue illumination. Some of these rings may illuminate red, indicating that these snails must be slotted in a specific place for their ability to take effect. We will cover those unique snails next week. You can queue up a space snail's next meal by feeding it one seed, then placing another seed on top of the terrarium. This second seed will remain safely slotted on top of the terrarium and will be consumed automatically when the effects from the first seed expire. Doing this will allow you to enjoy the benefits of the Galastropod special abilities longer without the need to stop and feed them again. You might have also noticed that there's a prompt to gaze at the space snail as well. Activating this prompt will pick up the terrarium and position it to give you a much closer view of the space snail inside. This will also introduce a new prompt allowing you to pet the space snail. I'm guessing there's some sort of telepathy thing taking place here, as it would seem you never actually physically pet the snail. But regardless of your telepathic abilities, each Galastropod will have a unique reaction to being pet. You can pet the snail over and over until you have had your fill of invasive species cuteness, at which point you can simply put the terrarium back down by selecting any area outside of the terrarium. Let's get the Galastropod Care mission complete. Locate any hazardous flora and dig it up to obtain seeds. Then place one on top of Sylvie's terrarium to complete the mission. When you do, Sylvie begins to emit light. A lot of light. Imagine what would happen if you had multiple Sylvies all happy and fed and emitting light. Well, you don't have to imagine it, you can make it happen. While inviting friends into your instance of the training simulation is something we will cover more in depth in a later course, if you do happen to have a friend join you, they will be able to interact with the mission log in your instance and use the recovery mission to reclaim Sylvie. You might think this would simply summon Sylvie from their current location and back to the landing pad, but you would be mistaken. Instead, your friend is able to summon an exact copy of Sylvie into your instance of the training simulation. If you invite more friends, they are able to use the recovery mission to reclaim their own copy of Sylvie as well. If your friends are thoughtful enough to ensure their copy of Sylvie is not contained in their backpack when they exit your instance of the simulation, then their copy of Sylvie will stick around permanently. So now you can indeed have a seemingly unlimited number of Sylvie, and if they are all fed at once, they can produce enough light to pretty much rival the sun. Before we wrap up today, let's collect your reward for completing the Galastropod Care mission, another QTRTG. And now let's spend just a moment discussing one other amazing characteristic that all snails share, but not all Astroneers know about. I alluded to this earlier when I mentioned a special vehicle from Astroneer Academy 106. That special vehicle is the gravity globe, and recruit, we are not the only ones who can operate this vehicle. If you didn't already know about this, prepare yourself because it's pretty incredible. Pick up Sylvie's terrarium and place it into the gravity globe. Sylvie will immediately begin operating the gravity globe. If you listen closely, you will hear them giggle with glee as they bump into things and even jump to try to get over obstacles. Space snails are not particularly good drivers, and they will run into obstacles, get stuck on tricky bits of terrain, and even drop down into any cave openings they may encounter. You don't have to worry about the snails driving off to parts unknown though, as they simply stop once they have gone a short distance away. Do be aware that they will resume operating the gravity globe as you approach, however, so you might be in for a bit of a chase. If you feel that a space snail has had enough time operating the gravity globe, simply get close enough to them and attempt to pick up the gravity globe. You will pick up the space snail's terrarium instead, which will cause them to vocalize their disappointment as their newfound freedom of movement has been brought to an end. And oh, would you look at that, recruit? This course has gone way over our usual ending time, so we are going to wrap up for today. I guess I got so distracted by the adorable cuteness of the space snails that I just lost track of time. Today, you have learned that something happened aboard the ISS Triton that caused Dr. Yo to jettison the Xenobiology Lab, and you began a mission chain to not only uncover the mystery of the Triton, but also become acquainted with Galastropods. We also learned that these adorable little buggers are actually an invasive alien life form, but they're so cute that we just want to pet them and feed them seeds. We can't help it. Next week, we will meet the remaining 6 Galastropods, completing the remaining Xenobiology missions in the process. Plus, we will discuss the limited information we have about Dr. Yo and the Triton, along with other space snail facts. Until then, I'm Brandon, reminding you to keep looking to the stars.